Welcome to our online release event. We are so happy to see so many people joining in online, and we are very excited because today we get to show you Omics Studio, a product that we have created because we feel that there's so much opportunity in omics data that is missed because there's not the right tools available for the researchers and for data scientists. So what is Omics Studio? Omics Studio is a bioinformatics platform, and it enables rapid hypothesis generation and also validation within the omics data. So it makes the process much faster, much smoother. And we created this product because we can see there's so many researchers who are using omics data, but they don't have the capability to, for example, use R or another coding program to extract all the information that is within the data. That requires a data scientist, but it's not always available in the research team. But also the data scientist needs a good tool to actually show the findings and communicate them to the general researcher. So this is a bit of insight into why we created Omics Studio. Now, what we're going to do here today is we're going to have a look at a proteomic data set. And it's a dummy data set, and it's quite simple. We have 3 different cell types from an HIV study, and we're going to dive into the data and see how Omics Studio can help build hypotheses and also test them within the data. Now, let's have a look at the data. And the first thing we need to do is, of course, set our study preferences. But before we dive into the data and the different workflows, any questions that you may have, please leave them in the comment section. The team will collect all the questions and will address them at the Q&A section after the walkthrough. But good, let's have a look at the study preferences. We're just going to do it short, but of course, a very important part is what kind of identifier do we want to look at. We're working with proteomics, so we could look at the UniProt ID, but for most that wouldn't make too much sense unless you're a hardcore proteomics guy. So let's just take gene names. That's easier to translate into what we're actually looking at. Then you have different thresholds and selection opportunities. Here we're just going to go with the default value filtration of 70%, and we're going to take all the samples and all the comparisons that we have. And then we also have some pre-selected preferences for enrichment analysis, and we're going to leave them on the default. And then I already selected what kind of database we're going to use for enrichment analysis. We're just going to look at Reactome as we don't have all day today, even though I could easily use it. But perfect, let's save the study preferences. And then let's first look at the expression view, because this is where we can just get a quick idea of what's the quality of our data and what principal component analysis, as we're going to dive into first, is, for example, a good way to see if there's any potential outliers in your data. So we're just going to start it, going to choose our expression data set, and if you have multiple expression data sets uploaded, then you'll be able to actually select more of them so you can do your comparison in between as you dive into them. There's also a filter to my list. I'll not go into details with that just yet. We're going to look at it later. It's a very nice feature that we really enjoy using when we are using our software. Then you can choose different contrasts. Here we just have sample groups, but this is also where you could have gender, age, or clinical variables that you want to be able to visualize when you're looking at the variance within the data. Then we have the samples to include, and of course we've pre-selected all of them, and we're just going to move forward with these. Then you will also be able to select feature metadata, which is basically if you have some features that you would like to add to your IDs in the tables later on, but we don't have any for this data set. So we're just going to process, and then we're going to use the scatter plots, because that's typically the plots used for PCA analysis. And if we don't choose it, we just get the table, which gives less information in my view. But as you can see, we have the tables, we have it for the loading plot and the score plot, and we have our scatter plots. And you see, first we're looking at the score plot here. So here we have the different samples according to the variance from principal component 1 and principal component 2. And what is very nice with this cell study is that we can actually see some nice clustering of the different sample groups. But this would also be where you potentially could see an outlier if we had a sample that was very long away from everyone else. And you can switch between different contrasts. We only have one, so that makes it easy. But you can also choose different principal components, because you might think, I only see something in the small parts of the variance, which could be PC4. And when you click, it instantly transforms and you have that data represented. So very nice and interactive. And you can download the plot, of course. If we go to the loading plot where we see the proteins that was used for the plot, then you're also able to select the proteins. So you can use them for example, a list, which we're going to demonstrate later. So that was just a quick introduction to the PCA plot. For the total identification overview, it's very similar. Here we're going to look at if there's any unique identified proteins. Going to take our data set. We're going to choose our contrast. We have some value filtration criteria we can then use as thresholds. Here we're just going to work with valid values in at least one group of 70%, meaning that there needs to be an actual measurement of the protein within 70% of the samples within at least one group. And again, we have the metadata that we could add if we had any. And then we're just going to run the analysis, choose our plots, and it's already ready. So let's go and see. So here we have the total identification counts, we have the intersections. If we have, for example, some proteins that are unique to a specific, group, but we'll just quickly jump to the PCA plot because this is mirroring TMT data, and we have all the proteins identified in all of the sample groups. So that's a good validation for that type of data. Then we have summed abundance calculation, and I already made the calculation here because we're going to choose the same filters, but we're just going to have a look at the plots. And summed abundance calculation, here we'll just have a look at the bar chart, and there you can see what was the summed abundance of the proteins within the different sample groups. And here we can see that condition Y actually seemed to have higher intensity readings than the other two comparisons, which might be relevant later. You can also look at it in the box plot view, depending on your preferences, but also they will provide a little bit more information. For example, here we can see that the spread between the samples is actually quite low, which is good to see in a proteomics dataset. Then we have the relative expression, which is basically where the data is, for example, log2 transformed when we are looking at proteomics data. And here a very used plot is the heat map, which quickly give a good insight into how are the expression divided between the different samples. And here you also just be able to choose mean expression if you want to look at the groups more in focus. And we can, of course, switch around with how we're scaling it, how are we flipping the axis, if we want to hide the identifiers, and so forth. And we'll be able to choose different proteins, and then they will come to our current selections, and then we'll be able to add them to my list, which is a feature we're going to dive into very soon. But good, that just gave us a good quick insight into the expression data that we are working with. You can see that it's of decent quality and seems like there's something going on between the cells. Now let's look at the statistics data from the actual data set. Good. Looking at the statistic view, here we can of course look at the statistical data from our study, and we can choose which kind of comparisons we want to focus at. For this, we're just going to include all, so we have something to compare, and then it runs. and we can choose the plots that we want to have a look at. And you can see the data is already ready. So let's dive into it. You can see first, of course, the table from the statistical data that we loaded into Omics Studio. Then we can get dot plots and volcano plots. And if we just look at the volcano plots, because that's something we commonly address when we're looking at statistical data, then we can see the first comparison, X versus Y. And we can see there's actually a lot going on. There's a lot of proteins that are downregulated and also quite a few proteins that are upregulated. And comparing that with the other comparisons, then we can see that X versus Z, there was actually more upregulations, and diving down to Y versus Z, then we can see here it's also dominated by upregulations. Now it of course becomes interesting to see, is it the same proteins that are upregulated and vice versa? And this is where my list comes into play. So we're gonna dive into that one already now. So what we want to do is, of course, see that on the left side we have a bar for my list. So this is where they're gonna be stored. But when we're gonna create one, we can do it already using the statistic view. So we can take a select box and then, for example, take a look at the most downregulated proteins in the X versus Y conditions. When we select them, we can see them over here. And then there's no my lists right now, but if we click, we can create a my list and say, this is the downregulated in the X versus Z. And we can make a small description. I'll not go into that. The name says it all. And then we're creating it. And now we can see we can select it and we can add to it. So now these IDs have successfully been added to this list. And then you can see in the right part of the screen that we can actually select from my list. So if we click here, then everything disappears because we haven't selected a list yet. But if we do select the list we just created, then we now have a volcano plot that is just providing these proteins. And we can see, as we saw, X versus Y, everything is downregulated. But if we go to the X versus Z, then we can see that the proteins that were downregulated are actually upregulated for some of them in X versus Z. And if we look at Y versus Z, then it's actually the majority, well, it is the majority that are upregulated. So, and this is just one of the ways to use it. Now you can see it's only showing 99 proteins out of the 18,000 data points that we have. But if we choose our list and just plug them on, then we can set it down. So just looking at those proteins. We can see it's still too many for actually getting a good insight into the different IDs. But here is a good feature. We can zoom, but we can also adjust the height of our plot. So for example, we could set it to 1600 pixels instead. And now we actually can see the different proteins and see what was the fold change in the different comparisons, which is localized at the bottom of the plot. And we can see there's also some size difference to the dots, which is corresponding to the log 10 negative p-value. So this provides us with some good insights and ability to compare between our different comparisons within the statistical data. So this allowed us to make a list. Let's, for the fun of it, just add one more list so we can use that later on. We also want, I'll say, the upregulated in the X versus Z. And then, of course, we need to select these. And it's fast and easy, and we've chosen the right list and add to it. Now we can use it later on. But let's dive into the enrichment analysis. Here we have two opportunities. We can do an overrepresentation analysis and we can do a gene set enrichment analysis. We are going to start with the gene set enrichment analysis and also start the overrepresentation analysis because that takes a little bit longer. So we might as well have that running while we're looking at the gene set enrichment analysis results. But first, looking at the GCA, here we can choose different thresholds. And for gene set enrichment analysis, it's of course best not to choose any thresholds because it's going to be represented in the ranking from the enrichment analysis anyway. So let's continue and choose the database. You can see from a pre-select, Reactome have already been selected, and that's also the one we want to go with. So we can just hit process, and while that one is going, we can start our overrepresentation analysis. That one already jumped into the selection of our database. We'll just quickly go back so you can see the filter we're applying. Here we are looking at everything with a p-value below 0.05 because we want to be exploratory. We could also add more stringent criteria, but for this demonstration we won't. But we'll choose the Reactome pathway here as well and hit process. And then let's go to the gene set enrichment analysis, which we can see is now done. We're going to look at bar chart and dot plot from the results. And again, we have the table with all the different values from the calculation. We have the opportunity to find different filters and put in different criteria for which pathways we're going to look at. Or we can just browse through all of them, but that's going to take a while. So let's jump to the bar chart. And as before, we're only showing 100 data points, as there sometimes are too many for us to be able to render at a decent speed and also for you to be able to actually see. Let's look at top 15. And as you see, instant filtration, and we can see which pathways are there. And more importantly, as we had our different conditions, we can actually see it from all different comparisons, so we can compare them. This can also be done a little bit faster by just going into the cross-group comparison. You can see right now we're just at the enrichment, which will give the individual enrichment results. But if we go to cross-group comparison, then we get it from all. And when we choose top 15, then we get a quick insight into how these pathways are behaving in the different comparisons. So we can see X versus Y seem to have quite a unique pattern compared to the others. And it's very clearly presented here. And you can click on the different pathways, and then you can see it actually goes into your selection box. You can exclude it if you want, but you can also see the related identifiers. So these are all the proteins that are related to that pathway. And here you will actually also be able to take a list and create it related to the pathway, which could be the one in focus, and use that later on. Just remember to push add to my list. We're going to use that later on. Let's quickly look at the dot plot. We have seen what it is before, but it's always nice to see it related to the results that we are looking at. And again, we can do as before, compare between, and we can do the cross-group comparison and compare them more directly in one plot. But good. Very short insight to it. And next up, we're gonna actually use some of the lists that we have created, and we're gonna use them in database search. Because here you're gonna get the opportunity to extract data from a lot of different databases to actually get more insights into the proteins that you have selected, or metabolized, or transcripts for that sake. So you can set a scope, what you want to be able to actually see in the different tabs. So here we have molecular identity. We want to see that. We also want structural and features and function interactions. And in theory, everything is interesting, but let's just keep it at these 4 and then say we want data from Reactome and we want it from clinicaltrials.gov and from the Human Protein Atlas. And yeah, let's take UniProt just because we can choose our lists, and then instantly we get some information. So, and we can go through the different tabs, or we can just look at everything. For example, we can see that from these proteins, there's only information from one protein from the Reactome. If we then go to UniProt database, there should be more, as is from UniProt identifiers. And here we can see much more data. And we can click to our focus point, which could be functions and interactions. So for the UniProt knowledge base, we, for example, can see what other proteins does the individual proteins react with. And you can see information from gene ontology, molecular functions, and so forth. Something that's also very interesting and information we'll get from the Human Protein Atlas is where is our protein, localized? Where have it been found in other data sets? So here we can, for example, see some seems to be very represented in the breast tissue, and some in the cerebellum and cerebral cortex, and so forth. So here you can get an insight to if there's some specific expression patterns to different proteins. And we just selected one of our lists. We can, of course, quickly switch between them so we can view the data that is relevant. And now let's go back to OR, because that should be ready, and it is. And again, we have our matrix with our data, we have our bar charts, same ability as we have with the gene set enrichment analysis, and just provides us a nice. So that was a quick demonstration of what you can do, how you can build hypotheses in Omics Studio, and how you can find information related to the proteins of interest, and also how can you do some light validation of them. For example, with the protein pathway of interest, we could also have just viewed that in our statistical information to see if it actually was relevant. So good. This was one of the many workflows that you can do in Omics Studio. And I'm sure you can think of several more. I can at least as well. But I don't want to drag you through a 4-hour session of all my different ideas of how to explore the data. In terms of omics types, we support proteomics, transcriptomics, and metabolomics. And if you're working with transcriptomics data, then it's primarily RNA-seq data. If you're working with a full transcriptome data set, then you need to filter down to protein coding genes in order for getting Omics Studio to support it. In terms of organisms, we are supporting humans, rats, and mice. And this is, of course, something we're going to expand on. So we'll cover all the most used model organisms, so everyone will be able to use Omics Studio to really dive into that data. And we are also going to expand on the plots that you see, so you'll get different types of plots, but they are all going to be as smooth, as interactable as you have seen here today, because we want you to not just be able to visualize the data, we also want you to be able to interact with it and really drag out what's interesting and use it in other functions and features. And we, of course, also going to add a lot of different features, but currently we have narrowed it down to what we think is the core features for really doing rapid hypothesis testing and hypothesis generation. So now I'm gonna just quickly brush up on my coffee and cough a bit. And then we're gonna switch to an interview with our lead developer and our lead designer. And in the meantime, I'll also have a look at all the good questions that you have left us in the comment section. So I'll be ready to answer these questions in 10 minutes time. So see you soon.
Now it's time for some interviews. First, with our lead developer. I'm Patrick from Omics Studio, and today I'm going to be taking you through some of the technical sides of Omics Studio and also discussing some of the engineering choices that we made along the way.
Security is a critical topic in Omics Studio. From a technical perspective, what were the most important principles you wanted the platform to be built around?
Right, so for a solution like Omics Studio, security is obviously always going to be one of the primary concerns. And so for us, we decided to host the solution on Microsoft Azure, and we're also following all of the best security practices recommended by some of the best security engineers in the world. And in addition to that, the solution is hosted in data centers inside the EU, so all of the data and everything is by default GDPR compliant, which we know is a very great concern for our users.
Why was responsiveness such an important engineering priority for Omics Studio?
Well, I mean, as a user, there's nothing more frustrating than having the software you're using constantly freeze. And what we saw among similar solutions is that when you're working with large data sets in plots, for example, anytime you'd select a data point, the whole thing would just freeze. And understandably so, because this is a very difficult thing to solve. And we also saw the same thing in existing plot libraries, that every time we had a large data set and we plotted it, then user interactions would just slow to a crawl. So we actually ended up developing our own plot library, and we spent months optimizing it to make sure that every single user interaction...
Plotting tools, and why was it important for you to build your own?
There were a lot of limitations. We looked around at a bunch of different existing libraries, and none of them supported the kind of features and plot types that we wanted. So we might have a plot library that did support, for example, upset plots, but then it didn't have the types of interactions that we wanted, like zooming, panning, selection, and so on. So in the end, we just decided, screw it, we'll make our own. And this also gives us full control, which is something that is also missing if we go with an existing library. So that's the primary reason we decided to go this way.
During development, what were the biggest technical bottlenecks you had to overcome to make the platform ready?
So I think for me, there's three primary technical difficulties that we had to overcome. And the first one is the fact that we're not using Shiny apps like so many of our competitors are doing. We're building this with proper web technologies like ASP.NET. And I could talk about the technical implications of this for hours, which would probably be pretty boring, but what it basically means for you as the end user is that everything's going to be a lot more snappy, everything's going to feel so much faster. And in terms of the future, we're able to develop new features however we please, and we can scale this thing out however we want. We essentially have full control, which is not something you get if you use Shiny apps or similar existing solutions. So that's the first thing. And then circling back to the plots, obviously when you're building your own plot library from scratch, you're going to run into a lot of edge cases. Like, for example, how do you calculate plot margins, and how do you handle clicking a point, something as simple as that. Like, all of these things in isolation may seem like trivial matters, but when you combine them into one big proper library, it actually becomes a pretty difficult problem to solve. So I'm personally really proud of the fact that we managed to solve all of this. And it's something that we've spent at least half a year optimizing and working on. And then finally, the last key difficulty in terms of the technical aspects is definitely the cloud infrastructure. So how do you deploy something like this to a cloud provider securely? And how do you build a foundation that you can continue to build in 6-12 months, for example? So thinking all of that in from the very beginning has been an extremely difficult engineering problem that I'm pretty happy that we also managed to solve.
When users work with larger and more complex data sets, what engineering choices matter most in making that experience stable and reliable?
So for me, that's the fact that we've built all of this in a cloud solution like Azure. And so right now we've got a queue system. So when you start your analysis, like your overrepresentation analysis, it's going to be put in a queue. But something that we're working towards right now is that we're gonna be starting every analysis in parallel, and they're going to be running on their own compute instance. And this means that you can, as a user, start, for example, a slow overrepresentation analysis, and then you can start a gene set enrichment analysis, and these two are going to run in parallel, so they're not going to block each other, which means that if you go ahead and start a third analysis, like some kind of expression analysis, then that third analysis might actually end up finishing before the other two. And you can then jump between all of these analysis results with just a couple of clicks, which is pretty unique, I think. So was that all of it?
Yeah.
Okay, so that was all the questions. So I hope this gave you a little bit of insight into the technical aspects of Omics Studio and why this solution is so unique. That was quite interesting. Let's see what our lead designer has to share.
I'm Louis, lead designer here at Omics Studio, and I've been asked to answer some of the questions related to our design choices.
Which considerations go into designing a bioinformatics platform?
Good question. And to be honest, there is quite a lot of different considerations when you design a complex platform as Omics Studio. First of all, most people think about the design, the colors, how everything should look and feel, which is of course also an important part. But there's so much more. How do we design the workflow so in Omics Studio? And to show you here on my screen and everything meticulously, in the sense that even the gaps between elements has been very finely defined, and we haven't just defined one view of things. Everything is mapped out very carefully, which ensures that everything has been thought through. But the other part is as well that we could go out early and user test with users, get validation to ensure that the workflows that we have designed actually make sense for the end user before starting to develop them. So yeah.
How do you ensure that the user experience remains consistent?
Well, consistency is key, and one of the things we learned quite early on, especially when we looked at other bioinformatics softwares and also interviewed their core users, was that the consistency is often lacking in those. Good example is that in one analysis module you have the button over here, and the next in the opposite corner with a different layout, a different text, which just adds to a confusion. And we want the researchers to be confident in the tool. And to be confident, you need to be able to learn it quite rapid. And again, have the things easily recognizable to minimize confusion. And a good example here is, for example, if we go to our statistics view and we briefly process it here. If we jump here to the dot plot, you can see, for example, with filters that they have a particular order. And if you had to jump between the different data analysis modules, you would see they always have the same structure. And the same goes for the buttons that are placed in the same position always. And that comes back to the consistency and making sure that it is easy to understand and follow, helping you and minimizing again the confusion around how to use the bioinformatics platform.
What have you done to make it easy and intuitive, especially for users who do not necessarily have a data science background?
Good question, and we have done quite a lot. It goes all the way back to the design thinking behind Omics Studio, and one of the good examples is if we go to the database search. Not necessarily all researchers know what databases to use in the specific context, and here we already have defined it's a proteomic study. We know it's a Homo sapiens. It comes from when we uploaded the data. So it already narrows down to the databases relevant for only those. And again, if I removed proteomics and Homo sapiens as filters, you can see now we have quite a few more databases available. So that is the first step where we can narrow it down. But it might also be that you have a specific focus in mind. And here we have also added the research scopes. And if I fold out the Uniport knowledge base, for example, here, and we say, okay, I'm actually only interested in the molecular identity, then you can see it helps you narrow down the scope to only that. And again, we could also say we are also interested in the function and interactions. You select that, and that's what then we are going to present to you, instead of having everything, which can be quite overwhelming. And if you jump to the database search here and select two lists, I just take it down and upregulate it from our dataset, you can see again here that you have the views, and you can easily jump between them and get the view that you need. More so, you can also click on those and get all the details if it's relevant. So that is just one small example of how we have made it super intuitive and easy for non-experts, especially non-data scientists and bioinformaticians, to get going with Omics Studio without any prior knowledge. This was a short glimpse into Omics Studio and some of the design criteria and thinking that we have put in to make the best possible bioinformatics software. Of course, there's much more to learn, so the team and I, we are super eager to get your feedback and learn how you're using Omics Studio, so we can iterate further on it, and it can only get better from here.
Turns out it is more than pretty colors. Back to Kenneth and your questions.
Good. And we are back at the Q&A session. So we have been looking at all the good questions that have been rolling in the comments to answer the best I can. And of course, you can always write to us and get more in-depth questions asked and answered. And also, should there be any questions that you don't see or remember today. But yeah, Patrick, let me hear what they have in store for us.
So the first question is, how does Omics Studio differ from other platforms such as Quaigen, IPA?
Very good question. I would say the main feature difference at the moment is the MyList. Because I say we integrated the MyList function because we want to make it fast to actually validate hypotheses within data, but also keeping focus. And also, Omics Studio is, in our opinion, more user-friendly.
And the next question is, in relation to transcriptomics, does Omics Studio support only bulk data or also single-cell data?
Only bulk data at the moment. We first want to ensure that the users get the most out of the bulk data, but then we also later on going to look at features so we can integrate single-cell transcriptomics data.
And the next question is it possible to access, inspect, or modify the underlying code in the platform, or are the workflows fully abstracted?
The users can't access the code. We want Omics Studio to be a non-coding platform, so we're not going to make it possible to access the code for different features and change it, because that also allows for, other people to make potential errors in the setup, and yeah, that's not something we want. We just want it to be non-coding and easy to use.
And next up, can I use MyLists to generate customized heat maps?
Exactly. MyList is exactly so you can isolate data within all the different features within Omics Studio, so you get some focused plots around your data. And heat map is one of them, but you can do the same with your bar charts. You can also do so you get all the box plots that are only related to your, the proteins or the transcripts and the metabolites within MyList.
Is it possible to upload more than one omics type and analyze multiple datasets in parallel?
Yes, you can upload as many datasets as you want and add them to your, your different analysis. At the moment, you can only run 5 datasets in parallel, just to not overcomplicate it, and also because it becomes quite computationally expensive and it will start to be a little bit slow, but you're allowed to do it, and it makes it so much easier to ensure that you are applying the same thresholds across datasets and make it so much easier to compare datasets in between.
And next up, when exporting a chart, what file format is it saved as?
It's saved as a SVG, so you can scale it as much as you want. There's no limitation, and it will always be sharp for your presentations and for your publications.
And next up, what types of plots will be added in the future?
There's long plots that we want to add. Some of the first ones will be the circus plots, where we can see, that's related to pathway analysis in the beginning, so you'll be able to see which pathways react up and downstream with other pathways, and also, of course, on the protein or transcript level, what kind of interactions is known in the literature. So that is one of them, but then there will also be violin plots and all the different other ones that we see common used in publications, so we ensure that you have the plots that you need.
And the final question, will it be possible to share projects and studies in the future?
Indeed it will, because that's also one of the things that we want Omics Studio to become. It needs to become a platform where you can share the dataset with your collaborators, both across, for example, European projects, but also if you want to collaborate a researcher with a data scientist, making iterations from the different steps that the individual have achieved. So, for example, it will be very easy to pick up on the my list created by the researcher for the data scientist, so that they then can perform some more advanced analysis of the data and some more validation. I think that will be all the questions that we can do, guys. I can see the clock is ticking. So first off, I just want to say thank you to everyone out there for joining us. I hope this session have given you a clear idea of how Omics Studio can be used for your research, and how it makes gaining biological insights faster and with more confidence. And yeah, we demonstrate a few different workflows, but there's a ton of them out there. So if you want to dive deeper into it and explore it, reach out to us. We'll happily give you a private demo where we can go into your specific workflows and your needs, or also reach out to us if you want access to the platform, or you just want to discuss some of the science in your project and see how do we believe that Omics Studio can help you answer your research questions. You, of course, also get an email after this event with a link to the video from today, and also with the voucher, so you can get your unique offer for Omics Studio. And yeah, that's it. Then I just want to say thank you all for your engagement, and looking forward to connecting with you and hear more about your research.
Omics Studio. You should try it out.